A very warm welcome to everyone. We, the following students, are going to elaborate on the topic RNA splicing. It was discovered in the year 1977 by the scientists Richard J. Roberts and Philip A. Sharp. By definition, we can define it as RNA splicing is the process by which the newly synthesized pre-mRNA, also known as heterogeneous nuclear RNA, is processed and forms the mature mRNA. hnRNA is processed in the nucleus and converted to mRNA, which then comes to the cytoplasm and undergoes translation or protein synthesis. In other words, it is a process that removes the intervening non-coding sequence of genes, introns, from pre-mRNA and joins the protein coding sequence, exons, together in order to enable translation of mRNA into a protein. There are two main steps in splicing. In the first step, the pre-mRNA is cut at the 5' splice site. In the second step, the 3' splice site is cut and the two exons are joined together and the intron is released. Splicing occurs in the nucleus either during or immediately after transcription. Splicing occurs in a series of reactions which are catalyzed by the spliceosome. A spliceosome is a complex of small nuclear ribonucleoproteins. The splice donor site at 5' end of the intron includes an almost invariant sequence GU. The splice acceptor 3' end of the intron terminates the intron with an almost invariant AG sequence. The branch point near the 3' end of the intron includes an adenine nucleotide which is involved in Larian formation.
Two types of spliceosomes have been identified, major and minor, which contain different types of snRNPs as shown in the figure. The major spliceosome splices intron containing GU at the 5' splice site and AG at the 3' splice site. It composed of U1, U2, U4, U5 and U6 snRNPs as shown in the figure. A number of proteins including U2 small nuclear RNA, auxiliary factor 1, U2AF2 and SF1 are required for the assembly of spliceosome. The spliceosome forms different complexes during the splicing process.
Complex E, the U1 snRNP binds to the GU sequence at the 5' splice site of an intron. Splicing factor 1 binds to the intron branch point sequence. Factor U2AF1 binds at the 3' splice site of the intron. Factor U2AF2 binds to the polypyrimidine tract. In complex A, the U2 snRNP displaces SF1 and binds to the branch point sequence and ATP is hydrolyzed. In complex B, the U5, U4, U6 snRNP trimer binds and the U5 snRNP binds exons at the 5' site with U6 binding to U2. In complex B', the U1 snRNP is released. U5 shifts from exon to intron and the U6 binds at the 5' splice site. In complex C, U4 is released. U6 U2 catalyzes trans-esterification, making the 5' end of the intron ligate to the A on intron and form a lariat shape. U5 binds exon at the 3' splice site and the 5' side is cleaved, resulting in the formation of the lariat. In complex C U2, U5, U6 remain bound to the lariat and the 3' side is cleaved and exons are ligated using ATP hydrolysis. The spliced RNA is released, the lariat is released and degraded and the snRNPs are recycled.
Here we see a RNA molecule with a single intron. Several signals exist within the intron that are used in the splicing process from the 5' end of the intron. This is GU, the A branch site, a pyrimidine-rich region on the 3' AG, the GU and AG sequences to find the beginning and the end of the introns. Splicing is initiated by spliceosome, which consists of several protein RNA complexes. The first step involves two complexes that bind near the GU sequence. The RNA is then looped and the three other protein RNA complexes bind. The final complex that undergoes conformational change. Introns are non-coding RNA sequences that must be removed before translation. The process of removing intron is called splicing. The intron is cleaved at the 5' GU sequence and forms a lariat at A branch. The 3' end of the intron is next cleaved at the AG sequence and the two exons are ligated together. As the spliced mRNA is released from the spliceosome, the intron branches are further cleaved.
Recursive splicing. In most cases, splicing removes introns as a single unit from precursor mRNA transcripts. This has been found first in a ultrabithorax gene of the fruit fly. Next is trans-splicing. Trans-splicing is a form of splicing that removes introns and joins two exons that are not within the same RNA transcripts. Trans-splicing can occur between two different endogenous pre-mRNA or between the endogenous and exogenous or artificial RNAs. Cell splicing. Cell splicing occurs for rare introns that forms a ribozyme, performing the function of spliceosome by RNA.
tRNA splicing. tRNA splicing is another rare form of splicing that usually occurs in tRNA. In the yeast Saccharomyces cerevisiae, a yeast tRNA splicing endonuclease, heterotetramer composed of TSEN54, TSEN2, TSEN34, and TSEN15 cleaves pre-tRNA at two sites in the acceptor loop to form a 5' tRNA, terminating at 2'3' cyclic phosphodiester group and a 3' tRNA terminating at 5' hydroxyl group, along with the discarded intron. Yeast tRNA kinase then phosphorylates the 5' hydroxyl group using adenosine triphosphate.
Importance of RNA splicing. RNA splicing facilitates the formation of multiple function mRNAs from a single transcript which code for different proteins. It helps in regulation of gene expression and protein content of the cell. It assists the evolution process by forming different combinations of exons and thereby making new and improved proteins. New exon can be inserted into the intron to create new protein without disrupting the functionality of the original genes.
The disadvantages of RNA splicing includes it has been suggested that 1/3 of all diseases causing mutation impact on splicing. Common errors include mutation of a spliced site resulting in loss of a function of that site, results in exposure of premature stop codon, loss of an exon, or inclusion of an intron. Here are the references that we have used. And thank you.